Hi, good afternoon and welcome, Dr. Barry Byrne from the University of Florida, Gainesville. I think most people are very familiar with Dr. Byrne. I'm Pat Furlong from PPMD, and we're going to talk about antibodies and gene therapy. I think this is such a difficult decision and discussion for patients. So, Barry, could you start off talking to us about gene therapy and antibodies? What do we have to know about those antibodies?
Sure. And thanks, Pat, for having me talk about this important topic because I think there is a lot of education to do in the community. So gene therapy products are made up of two principal parts. One, the coating or the outside part of the virus is made of a protein. And inside of that protein coat is the genetic information, which is used to that delivers the therapeutic gene. And in the case of Duchenne, these are various forms of dystrophin or microdystrophins that restore the function of the protein. And so there are two components, and that one component on the protein part is present at the time it's delivered, and much like a vaccine, causes a response universally in patients who receive gene therapy products that we believe have believed in the past would immunize, in effect, them against further treatment. We now know that there are probably ways to prevent that immune response, which is not what we generally want with immunizations, but in the case of a gene therapy product, we want to preserve the ability to either give additional doses of that product in the future, and I think we'll touch on that. But that's the nature of the immediate response to the gene therapy product is that part of the virus that's borrowed from AAV in particular, if we're talking about AAV vectors.
Barry, then, in each of the protocols for all the companies that are either conducting gene therapy trials, or now we have an approved product, Sarepta's Elevidys. So they're asking for basically a test in Sarepta's 1 to 400. Can you explain what that means? And what if I'm 1 to 410? Does that eliminate me? And what relevance does
Yeah.
that have?
A great question. So each sponsor has to develop a test method. So it's called an assay. And these assays are used to measure the antibody levels, and they're specific to the product. Even though there may be several types of AAV-RH74 products in the future, the one that Sarepta developed, for example, is linked directly to Elevidys, and it's called a companion diagnostic, and it is approved as a separate medical device, in effect, because of the method that's used to detect the antibody. And this is done to try to ensure that it's reliable and reproducible every time the test is done. And what Sarepta was able to do was show that using the assay that they had developed for the antibodies against RH74, that in the non-clinical studies that enabled the clinical trials to be done, that there was no impact of antibodies on the efficacy of the product up to this level of 1 to 400. But as you point out, 1 to 410 is very close to 1 to 400. And does that really matter? And it, in fact, probably doesn't matter. But the way the test is reported, it's either pass or fail. And we don't always get, in some cases we do, for Elevidys we do, but in other products, we only learn whether it's acceptable or not. And that doesn't help us understand whether it was just over that limit or effectively zero. The report looks the same. So there's a lot to be done to standardize those results so that people can understand better how it can be addressed and whether there is a way to mitigate that antibody, even if it's for another product, which there likely will be many different products, and specifically, ones that even have greater effects than the current ones, that may be related as proteins, but we'll have new tests for those that are not the same as the tests we have today.
So, and the tests are these tests generalizable? So if I fall outside the protocol of Sarepta, for example, and I'm at one 410. Is that generalizable across? Could I still be included in a different protocol by another company?
Sure. There are two types of tests, and a one that tests the ability of the virus to infect cells that are representative of the ability of the virus to bind to those cells, and one other kind of test, the total antibody test, looks at all the antibodies in a reaction that binds the vector to just the antibody itself to the vector protein. Those are two completely different assays that are not, the numbers are not transferable between them. And in some cases, for example, the Pfizer product in its current studies uses this neutralizing test to bind to cells, but all the others use the total antibody test. And that was a sponsor's decision. They felt that that more accurately represented the biology of what the antibodies might do in the body. So there isn't a common platform, unfortunately, to directly answer your question. We think that there are ways to actually do all of the various existing vectors in one test. This would be more likely to give some insight instead of having one lab do test A and another lab do test B. If there were a way to what's called multiplex or combine the tests against all the commonly used vectors at once, this might be valuable to help patients understand whether the results are relevant between different products.
That would be amazing because, as you know, patients want to, and their families want to know what they might be eligible for so they can make decisions about access to clinical trials or access to treatments once they're approved. So I think having a test such as that would be really incredible. But there are patients who, as you know, have antibodies to the virus, and it feels to them, once they get that report, that it's the diagnosis all over again. And it's heartbreaking because they feel like there aren't any other options then, because I think we've learned in our preference studies that having dystrophin on board, whether that's an expressed dystrophin through antisense or through a gene therapy product, is reasonably likely to provide benefit. So once you get those results, it is incredibly heartbreaking. So what can we do? And do you see a day soon? And I don't even like the word soon, because soon for a patient and family is tomorrow, but within a reasonable limited period of time, a way to address those people, those individuals that fall outside the window of a gene therapy product.
Yeah, we really, I appreciate your recognizing this as a critically urgent problem that families face, and particularly with access now to a commercial therapy, we are encountering individuals who are not eligible, who are approaching their 6th birthday, and as you said, this is now, we have to do something about it right away. And thanks to the support from PPMD, we're able to initiate a study with the help of argenx to use a new product that's used in myasthenia gravis to try to lower antibody levels. And the initial evidence we have from both the non-clinical studies and some data from argenx is this will be helpful up to a certain level, and we have to determine in some volunteers for that study, plan to start early next year, how much we can lower antibodies using that approach and what other approaches might be added onto that. And that's our immediate goal, really started a number of years ago in one study that actually enabled us to show that we can give multiple doses of AAV. We're building on that in the context of Duchenne and understanding how can we address pre-existing antibodies and how can we attenuate the antibody levels after the exposure. And that information is described in a paper which has just been published in the Journal of Clinical Investigation. So we can provide you a copy of that to distribute to the community.
That would be amazing, Barry. So I think this is music to our ears. So just thinking about a timeline and going forward, and I'm not asking you to give me day and month and time, but you would see in the relatively near term the ability to treat people who just fall outside the window of antibodies, and perhaps in a little bit longer term, the ability to retreat or redose patients who've been in a gene therapy protocol. Have I stated that correctly?
Yeah, so in the paper that's just been published, there were 13 individuals that out of the 38 we studied where the antibodies were prevented altogether across a variety of ages, including into young adults that received a treatment for Danon disease. So these weren't all Duchenne patients. This approach has not yet been used in Duchenne, but we have reasonable evidence now from small to large individuals that receive these varying doses of AAV that we can prevent the antibody response altogether. And if those medications are used prior to treatment, the antibody levels also slowly decline. And that approach plus this Argenx product may be one way to mitigate the antibody responses that exclude people from treatment today.
So, and so one of the questions will be is if you're trying to prevent an immune response, does that in any way limit the effectiveness of the therapy as you're delivering it?
No, to the contrary, actually, what we've learned is it actually can as much as double the expression level. So this is going to be a really important aspect, particularly to gain the maximum benefit from existing products and future ones. And there'll be even more specific ways to do this than the approach we're using today is a very general approach that lasts several months. I think there are even more targeted therapies that could be applied in the future. But for now, several biopharma companies have adopted this approach for their commercial development, not in Duchenne yet, but as I said, this will, I think, lead the way to other drug developers to consider this approach in Duchenne. And we may have the opportunity to do this in some patients who receive commercial therapy, and we'll be able to show that if in fact the effect of the drug is improved because of the limitations of the immune response, this is going to be beneficial to the patient community. The same concepts apply to the transgene immunity we've seen as well, and we think that's an important question also.
No, I agree. That's an important question as well. But I think, I think the community's fundamental concern is how can I get gene therapy? How can we lower the antibodies if I fall outside the window? And then the percentage of the patients now being treated with our first generation product and certainly others to come along the way is going to be, as I grow, as I get bigger, can we preserve what's there, which is preserve and protect that muscle is the goal of, I think, every parent and every family.
Absolutely.
Well, I can't thank you enough for your work. We are thrilled to be supportive. We'll support whatever you can do to really transform these lives as you're doing now. We're thrilled about gene therapy and hopeful for a very bright future.
Well, thanks, Pat. Really enjoyed the conversation.
Thank you, Barry.